Hi guys, welcome back to episode 2 of series 2 of LifeSci AI, the podcast series. And continuing the theme of twos, we have our first two guests in the podcast today, and we're really thankful to have another application of AI being used in the life sciences. As everyone knows, that series 2 is all about broadening everyone's horizons and seeing what other applications we can bring to you. And today we're going to be focusing on molecular imaging and gene therapy with an incredible biotech based in Paris. Now I'll let them go into a bit more detail about who they are when I sort of pass over to them. But we're really thankful to have both Mark Lynch and Andrii Kulakovskyi. I hope I pronounced that right, Andrii.
It's fine, it's fine.
From Genomic Vision, based in Paris. So thank you very much, guys, for joining me.
Thanks,
Thank you, Nick.
I don't know who wants to go first, either Mark or Andrii, and just sort of giving an outline of your role in Genomic Vision and what you guys kind of focus on as well.
Sure. I can go first if that's okay, Andrii.
Yes, please.
Yeah. Okay, so as mentioned, Nick, my name is Mark Lynch. I look after Genomic Vision's commercial and marketing department. I've been with the company since March 2021, so actually not the longest time. And before that, I was actually working in various other biotech companies based in Europe, but also in the United States. And I joined Genomic Vision because the technology is so compelling. It's able to reach a level of resolution that other molecular methods just cannot do. And so it means that it's a technology that, and a company actually, that sits right next to all the big players in the molecular genomic space, such as next generation sequencing and such as real-time PCR. But it provides that resolution that's not quite possible by those other methods. And so what you'll see, hopefully, in this dialogue between Andrii and myself is that it's a method that provides a view, hence Genomic Vision, into the biology of genetic diseases that isn't always possible by other methods.
Awesome, cool, thank you. And Andrii, what do you get up to at Genomic Vision?
There are many ways how you can use artificial intelligence. And maybe the best one from my perspective is to use it for life science, when you can basically help to cure some diseases, detect them, cure, and invent new drugs to potentially save people's lives. And this is the base thing you can do as an artificial intelligence engineer.
So you're the head of BioIT, a new department?
That's correct.
Genomic Vision, and you both joined very recently. So I guess why did you both join Genomic Vision? You sort of intimated it slightly there, Mark, from your side, but was it a company that you guys had seen on the market a lot, or did someone come to you about it, or was there a step change? Because you guys Genomic Vision been going for about 10, 15 years. So was there a step change that you guys thought, oh, we need to get on board with Genomic Vision now?
Yeah, I mean, again, maybe I can start. So, I mean, I actually joined, we had a changeover of CEO last year, I think it was. And actually, the CEO, Dominique Remirou, took over from our founder, Dr. Aaron Ben-Simon. Aaron is still here and helping us in the company, but Dominique is here to help to strengthen the kind of commercial execution of the product. So in other words, building products from end to end, from start to finish, of course. And one of the reasons I joined is that Dominique and I actually brought new technologies to market previously in our previous roles. One of the things that we did was establish the single cell omics market. For those that don't really know what that is, it's when you take a single cell and you can sequence and get all the transcripts from that single cell and understand any changes in cancer cell clonality that is impacting caused by the cancer or caused by the drug. And so we established that previously. And to be quite frank, we want to relive that experience at Genomic Vision. It was very exciting, and really the innovation is key, and that's what we want to do here.
Yeah, so that's very similar to the episode previous with Quantum Surgical. And if you guys haven't listened to that, go back and listen to it. Lucien Blondel had created a couple of startups, and they got bought by various other bigger medical device companies. And now it's almost like he's getting a band back together at Quantum Surgical to build again. So
yeah, it's so much fun. It's so much.
Yeah. And what about you, Andrii? Because I know you we were just chatting before we started recording about your PhD and what you did previously. So I guess, yeah.
Okay. So actually for me, it was a bit similar to Mark. So similar in terms of motivation. I started my career in aeronautics, where I was working on the physics-based computer vision. Then I moved to the telecom sector, when I was using also artificial intelligence, but for different purposes. But at some point, we had a very detailed discussion with Aaron, the founder of Genomic Vision, and we were discussing about the challenges and potential future impact that company may bring to the life science. And the possibility to bring this product to the market, to bring this technology to customers is something incredible. And I also want to live this experience.
Yeah. When moving across from the aeronauticals industry, what was the same and what's a little bit different? From
telecom and
yeah, so from your PhD, looking at, because we're chatting about your physics PhD, but it was within fault detection in aeroplanes. So someone who doesn't like flying, my anxiety in life is in your hands, Andrii. So what is different? I don't know about the data or the restrictions or the approaches or techniques that you were using and that you are now using perhaps in life sciences.
I will try to draw some parallel lines. So when I was working in aeronautics, I was developing with a big team of other experts. I was working to develop basically a system which would survey the aircraft and to predict its remaining lifetime. We were implementing the set of sensors into the structure of aircraft and then collecting the signals in order to build the images, to analyze them to predict the remaining lifespan. And there are some parallels with the life science, because at some point you need to take a huge responsibility on your product, because at the end your conclusions directly impact the estimation whether the plane can land or can fly, or we should do the maintenance immediately, or we can plan it in advance. And this is something similar what we do here. We analyze DNA, we try to see if everything goes well. There are some mutations, so possibly we'll have a disease. If you do the drug discovery, does it work well? So in terms of responsibility, in terms of prediction accuracy, they are quite on the same level.
Yeah, I see. So it's like that. I don't want to say life or death, but the responsibility of your work, then that you find that very similar, and that was sort of the motivation really, and you saw that quite an easy crossover in that point.
Exactly.
Awesome. Okay.
And at the end, you still work with images.
Yeah, exactly. They don't change.
They don't change. Maybe different, but at the end, it's an image.
Yeah, for sure. And for yourself, Mark, were you always going to go into biotech, into life sciences, or was it a bit of
Yeah, for sure. That was the only thing that I was ever passionate about at high school, for example. And then, of course, went into university and studied microbiology. And and you have a choice when you're in academia. You can either go down the route of getting a PhD and going through the academic channel, or leaving and trying to go into a more commercial environment. And that's what I did. I went to a more commercial environment, but actually I didn't find that all that interesting. What was actually interesting was bringing new technologies to market and understanding the technology, not necessarily the technology, and being able to convey that to new people in the field, being able to establish relationships with key opinion leaders and be able to advance their science. We bring the technology, but ultimately it's our customers that advance the science.
Yeah, and I guess that's kind of important to note about Genomic Vision is you guys are an enabler, as it were, for the scientist. and for the research. So I don't know, Mark, if you want to go into a bit of detail about sort of the 3 areas of genomic vision, because I know that's something that you're very passionate about and wanted to explain.
Yeah, sure thing, Nick. Yeah, and we can pretty much do many other applications, but there's really 3 key applications that we focus on as a company. And actually, these applications tie into different levels of enablement for our customers. So the first kind of application that we work on is actually based on something called replication, which is really looking at cells and understanding how those cells divide and what impacts the ability of those cells to divide. For example, exposure to various environmental factors or exposure to drugs during cancer therapy. So our replication assay is really used as a research tool. To understand potential drug targets, to understand the mechanism of how the drugs interact with the cells and repair those cells. And so we work with academic customers in universities, in hospitals, and even in some biotech for that particular application, to be able to understand the replication kinetics within cells. The other application area that we focus on as a company is to provide a tool to pharmaceutical clients. A lot of these pharmaceutical clients these days are studying how cells can be edited to create vaccines. Of course, in this time of COVID, that's a big focus. How those cells can be edited to understand, again, different drug targets. And they use gene editing technologies such as CRISPR-Cas9 to do that. But there's a very critical part of the workflow by which you need to be sure that the edits that take place are in the correct place within the genome, and to make sure that it has no adverse effects. And so a lot of these pharmaceutical clients, for example, Sanofi, use the technology to be able to understand that, to use it as a quality control method, to be able to know that the edit that has taken place is in the correct place within the genome, and that it has no adverse effect. In other words, it has the desired effect for the drug development. And so that's another big aspect of our molecular combing technology. And the final one, which I leave to the last because it was actually the founding principle of Genomic Vision, is a diagnostic, using the technology as a CE-IVD test for rare diseases such as FSHD. And that application is leveraged across many of hospital labs in the clinics, for example, in Latimon in France. And we use that as a diagnostic test. And so that's another big sphere of how our technology is used.
Awesome. And thank you for taking us through this. I guess for me, the one that picked out, well, the last one I just want to touch on in a second, but the second one about sort of genomic editing, does this work in the workflow for the drug discovery? So does this make the research more efficient because you're getting it checked before it goes through to any trials or anything through there? So it's just making it a more efficient process. That's
exactly right. That's exactly right, Nick. And so it really does increase the efficiency. And any pharmaceutical client or customer or potential customer who is performing, drug discovery studies or actually vaccine development, and have to do this via conventional means. There's actually an old method called Southern blot that can actually be used for this, but it's not quantitative and it takes a lot of time to actually do it. Our molecular combing technology is faster, more sensitive, and it gives a quantification result. It means that you know precisely if the edit is in the correct place or not.
How early is this fitted into the process?
It's pretty early. So normally the pharmaceutical labs would have their cell lines in kind of cell development factories. It's essentially like a cell production site. Those cells are all cultured and cultivated. And it's actually right at that process where you would test that. You would insert for example, the gene edit, and then from that stock of cells, you would then apply the molecular combing technology.
Oh, I see. Because I guess if it's very late on in the process, it becomes very inefficient. So you want to get as early as possible.
Yes.
And the rare disease point is very interesting as well, because it's something that people within SciPro look at as well. And we actually have a blog series called Rare. So we do look at a lot of supporting a lot of the rare disease and orphan diseases clients across Europe as well. So I guess what has been the impact within that piece for rare disease? And you mentioned a specific one there, and for the diagnostic tool that Genomic Vision have been working on for a number of years.
Yeah, I mean, the impact is pretty clear. We are the market leader in FSHD, which is a particular type of muscular dystrophy. And our tool, our technology to be able to do that has been validated as a CE-IVD. So it means that the wet chemistry required to run that test is CE-IVD. The hardware, the FiberComb system and the Fiber Vision system are also CE-IVD. So it means that the hospital labs that choose to adopt that technology have a full solution that is validated as a CE-IVD. That's very impactful. Now, to answer the other part of your question, Nick, related to what impact does that actually have for those hospitals? If that technology does not exist, they are again using old type methods such as Southern blot analysis. And the best way to think of a Southern blot analysis is almost comparing analog to digital. Analog kind of readout of a kind of band on a piece of lab paper versus digital readout where you actually get a value, a quantified value, but you also, to Andrii's point earlier in the podcast, get the image of the DNA. So you actually see the image of the DNA of that fiber so that you can understand if that patient has got the insertion that contributes to the rare disease. And that's not possible via the analog-based approach. To answer in a short way, it's very impactful what we provide for that market.
For sure. And it's fascinating that you draw the analogy of digital and analog, the way you look at it, because I remember when we had Mark Goldfinger from Perspectum Diagnostics on the podcast, he was talking about their digital pathology product, and he explained it in the exact same way. It's almost like going from, you're getting the lab from a typewriter situation into a computer situation, and everyone knows the difference between how good a typewriter is versus a computer. So it's a really good analogy to use, and it's interesting that you worked on that. But I guess I wanted to delve into now the use of AI with Genomic Vision and how you guys are utilizing it. And I guess I want to sort of bring it back into why has Andrii been brought in, and why were you so taken aback, Mark, by the sort of the technology and really brought in to take a new product to market? I guess if we go for the very early stages to why this sort of came about within the business, perhaps.
Okay. Yeah, maybe I'll touch on the start of that and then I'll hand over to Andrii if that's okay for the rest of it. Look, it's the same old story with biotech, honestly. Biotech companies are very good at creating the wet lab part. They're all generally wet lab scientists, meaning that they all came from a lab bench, pipetting samples into tubes or doing various other types of assays, whether it's assays and so on. The problem with that is that you always leave the dry part, the analysis part, the analytical part is always left to this kind of old-fashioned Excel sheet trying to look at data and data together. That is always, and it's really unusual to have a solution that goes from sample to answer, where the person doing the test, doing the experiment, is able to basically run it like you would run a mobile phone. And so what we're trying to do with our BioIT function is to make the back end, the dry end of it, as good as the wet end. And that's really where Andrii and his BioIT team come in. Andrii?
Thank you, Mark. Yes, just Mark mentioned the image processing part steps right after the wet part in order to complete the solution. So we have we cover the whole cycle from lab to the very end, the comprehensive report, which explains what you have done, what are conclusions. And here we have several domains where we use AI. It's image reconstruction. Then we have the detection of GMC, genomic Morse code, and segmentation, and then we have data analytics. And we are using a fluorescence microscope.
Yeah.
So basically, when you acquire the signal, a raw signal, it's a monofrequency signal. And at the end, what you want to reconstruct the conventional image that you can basically see on your laptop and understand. So first of all, what we do, we scale each of the monofrequency component, and then we fuse the stack of them to build the image, which basically reveals the DNA and the GMC code. Once we have built the image, we start processing it to detect the fiber, first of all, and then GMC sequences on it. And here we rely a lot on the convolutional neural networks to do the RE detection and then do the segmentation. So basically, each pixel inside the RE is assigned to the specific GMC signal. And once we complete it with this, we can do the analysis. But here's the key problem, or rather challenge. The typical cover sleeve we're processing contains 3 to the power of 10 pixels. So it's like 30 billion of pixels to process on each assay. And you can see there's a huge amount of data.
Yeah.
And if we deploy the convolutional neural network on the accelerator, we can reach not only the human expert accuracy, but also the fast processing speed. And what our ultimate goal is to replace the human expert in DNA image analysis and interpretation with automated detection system, which would be as much accurate as this person.
Yeah, I see. So on the use of CNN, is it the fact that you're layering the images to create the one DNA? Does that make it easier for you to manipulate and have a very nuanced layering of the CNNs for that particular DNA? Do you attach one CNN to one layer of the DNA and then submit it through, or do you combine it and then run one CNN through all of them?
Once image is combined,
yeah,
it's the entire image that goes into the CNN. So basically, CNN is intelligent enough to understand what is the fiber and what are the GMC sequences on it.
Cool. And how bespoke are the CNNs that you're working on? I guess you can't really, go up and grab them from, TensorFlow library and, So I guess what you're doing is so bespoke and cutting edge. You sort of writing them from scratch, or how's that?
Yes, indeed, we we do not copy paste the code. It would be such efficient. So what we do, I will start from the perspective and then we'll go back to details. So Genomic Vision invests heavily into BioIT, and we have the capacity now to do our own fundamental research on the CNNs for genomic Morse code detection. And this fundamental research lives with the company, and we derive the specific, let's say, sub-CNNs for a specific problem to solve, for a specific assay we would like to detect.
Right.
And that's why the question comes back to the fundamental research in AI. So what my team does, we basically split the work in 2 directions. First one is the research, and the second is the production part. But they are tightly connected.
Okay. So are you kind of creating your own bank of library of CNNs that's going to be very much for your guys' use then?
We do. We do create our own.
That's so cool. And then I guess the one challenge that I always hear is very challenging is, getting it from the research and productizing it or put it into production.
It's very different.
So what challenges are you finding, and maybe if there's some solutions that you want to share, then if you want to, and how detailed you want to go, but how have you found that? Because and has that, as a follow-up, has your PhD been quite real world focused, going back to what you're doing with plane fault detection? has that helped you understand what's needed to productize these algorithms?
My PhD was a wonderful experience, and I'll explain why. So the main objective here at Genomic Vision is that we not only detect the GMC on the single cover slip, but we are sure that this detection accuracy remains constant for many others. So basically, we reach some level of, let's say, confidence in analyzing the whole set of assays. And this is very different from research when you can basically run your small neural network on specific data set, which is well calibrated, which never change, and obtain very good results. You can publish a paper, right? But real life is that your images are always different. The noise is always different. The shape of the signal can also change. And where my experience from PhD comes in, that when you do your PhD, you really go into the very, very detail of your solution, and you basically need to understand every corner case. And this ability, that's why I really like to work with people who did just a bit of research in their life, because they can go into the very depths of neural network and search the key reason why at some point the neural network works perfectly and at some point it fails. And basically the very difference between the production and the research is that we need to fix all the issues in the productions that do not appear.
So for you, it's a mindset. You know, it's that mindset that you find it's been successful.
Correct.
Fascinating. And then with the research that you're doing, I know that the impact you have is within degenerative diseases and research around those. So we touch on different areas of degenerative diseases, anything from like very early stage neuroscience, and and how the brain is affected, and is it genetic or is it built up over time and stuff. So I guess where does Genomic Vision fit in to, on a wider scale, the degenerative diseases drug discovery research? I don't know who wants to take that.
Andrii, please chime in as always.
Thank you.
I mean, by definition, we are really looking at the genome maintenance, right? By definition, that's what we're doing, right? And what we mean by that is that we look at the replication, in other words, the replication of cells, how they divide, but we also correlate that replication and how those cells divide to what impact it has on the chromosome. And so by looking at that in a kind of dual mode, what we're really trying to do at Genomic Vision is understand genome maintenance. And what that provides insight to is diseases like Alzheimer's disease, all forms of neurodegenerative disease, whether it's early onset, whether it's late stage. All of our customers are interested in that, and that's a key aspect for us. The other key aspect for us at Genomic Vision is how that impacts cellular health, right? And what we mean by that is we've got this great technology that can understand why and how and the impact of cells dividing based on environmental factors, based on drugs that are being used for chemotherapy and cancer. But what we also want to understand is what effect that has on aging, not on cosmetic aging, but what impact that has on the aging body. What is going to happen to you in 15 years, 20 years? And that's a whole other market of cellular health. And that's something that at Genomic Vision, using genomic maintenance, we really believe that our technology could fit and provide key insights to that other technologies simply cannot do.
Oh, awesome. So it's very much the aging of the gene that you're looking into and tracking. So how would you track that from somebody, like on a really like layman's basic term? Say we've got like an 18-year-old, would you go in and take their genes out and then analyze it, and every 5 years you would do that? Is that where we're kind of heading to, or is there a different solution to that?
The great thing is that governments all over the world have already funded studies to do this. So there's there's biobanks that exist in every single country, and various consortia that run these biobanks. For example, in the UK, we have the UK Biobank, okay, where these samples have already been taken. They're sitting in freezers at -80 or in cryopreservation if they're cells. And so the technology really works in being able to go back to those samples and look at them over time and see how those samples have changed, over the years. So it really is part of an ongoing process. And really one of my roles at Genomic Vision is to make sure that the company, that the sales and the marketing team are engaged with those biobanks to make sure that our molecular combing technology is being used to study such research.
Yeah. That's awesome. So I guess not going into our GP every 5 years, getting our genes checked and coming out. Maybe, I mean, you never know. Someone, we talk about things now where we're getting into a cloud-based diagnostic systems for strokes. Like, who would have thought that? Like, Google Health released their first cancer detection mobile device. Like, it's crazy. So maybe in 20 years' time, we'll be having a conversation where we can analyze our genes on our phones or our next device, whatever it happens to be of that time, something very Iron Man based around that, Tony Stark. But I guess also I'd be really keen to know, because I know you guys are really focusing on, and Andrii touched on it, is the automation of the image analysis, taking it off an expert. Pretty much, like not like taking over someone's job in a way, but looking at how can we shorten the research timeline by reducing the analysis time needed and also the training needed for that specific person. So I guess I hand it over to you guys just to explain what that essence is and how you guys are looking to achieve it.
It's very important to automate the system if you want to become the enabler at the very basic of things. And so our technology becomes this enabler for other customers, partners. And the maybe the main point here is to say that the artificial intelligence is very good on the specific domain of application. So if you can basically define the boundaries where you apply the AI, you can reach the tremendous results. That's what you see, for example, on the neural network, doing classification, doing the image restoration, doing, video, like super resolution, what Netflix does, for example. And it's pretty much the same mindset that we have in Genomic Vision. So we're trying to approximate, and we don't try, for example, to, let's say, to take the job from someone else. We're trying to approximate to some specific skill of the expert that does the image editing, and we completely automate it. And basically, we not only developing the core algorithm for image analysis, we also developing the web application where each blocks of the image processing are synchronized and are integrated into the web application. So basically, we provide the whole solution where basically the scientist needs to simply click a single button to run everything from the very beginning to the real report, the comprehensive report explaining what you did. And this is about the automation we do. For image reconstruction, image analysis, data analytics at the end, comparing the control groups with the testing groups, and doing some predictions on the next steps.
A question for you then, guys. Are you considering using the cloud as well in what you're doing? Because I know there's one project that are utilizing the cloud to help the repetition of research. So somebody could use a device, run a piece of research, it's an image analysis system in the University of Manchester, for example. Then they upload their analysis, they upload their report onto the cloud system for the product. Then somebody in University of Sydney can take the exact parameters, the exact conditions, upload it onto the device. It runs it, analyzes it on their data, and then they can combine, and they can talk, and they can analyze and compare and contrast. So is that something that you guys have looked into?
This is very good question, Nick. Idea is wonderful on its basis.
Yeah.
But, and we actually work on it, honestly. However, today, at current communication and bandwidths with, for example, 4G technology, it's not really possible.
Yeah.
So we're starting to look into the 5G, how we can adopt this technology to really put everything in the cloud to do it immediately.
Yeah.
That's why we are closely working with different partners.
Yeah, like you said, how many pixels is it per picture?
Exactly. We have 30 billions of pixels per image.
Yeah, you'll be waiting there for ages for it to upload. So.
But in the same time, we're working on the compression algorithms to basically compress the data and make it much more easier to transfer.
So that will be the key. If you can harness the 5G interconnectivity with the compression algorithm.
With the compression, exactly.
It'll make it a smoother process to potentially.
And then we can rely on the full cloud solution without any delays, any lags, and it will be wonderful.
Yeah, we'll see how you get there. See how you go. But that was fascinating, gents. And we've got maybe a bit of a scene to the future as to where you're going. But I don't know if you guys wanted to touch on actually the future for Genomic Vision, because I know you guys are really going places and some pretty big goals in the next 12, 24 months. So I didn't know if, Mark, you wanted to give like an overview of where people listening or watching this can hopefully see you guys in the next 12 to 24 months.
Yeah, sure. So I guess the most important part is really to stress the BioIT part. We really invested heavily in BioIT, and seeing some of the outcomes before the end of the year is very important to us and our customers. And so one of the things that you'll see on our website is that we announced our Fiber Smart, which is actually our first artificial intelligence-based solution for automated image analysis. And that is going to be, if you like, the stepping stone to get us where we want to go. And I don't want to go into too much detail for that, right? Rise into what we actually want to do. But certainly I would say, Nick, that looking at the Fiber Smart and taking a look, actually, for listeners, you go to our website, you'll actually see the press release on it and what it enables for that type of replication analysis that we spoke of, for the type of diagnostic analysis for rare diseases like FSHD. That's a critical element. And the other kind of element to it is making sure that we are, as a company, really focused on things like genome maintenance, making sure that we are the company that the community come to for being able to understand structural variation in the genome in an automated fashion, and linking the disease effect to the aging process of cells. That, I think, is critical. And that taps into a whole range of different applications in pharmaceuticals, in research, and of course, in diagnostics.
Yeah, no, for sure. It's exciting. And so we will all probably be watching with bated breath, and hopefully the Fiber Smart gets the sort of, I don't know, the traction, or it proves that you guys, and validates the work that you're doing, and you can take the next steps moving forward. But this was fascinating for the first series 2 in episode 2 and all the 2s. It was really good. And I guess, I don't know if you guys wanted to touch on how people can reach out to you. There's probably some discussion points that people may have. I don't know if LinkedIn is the best way or what for you guys.
Absolutely. Our LinkedIn channel is manned by the marketing team. So if anyone wants to reach out via LinkedIn, they can. Both Andrii and I are individually on LinkedIn. So do that as well. That is not a problem. And if that isn't what people want, you can also email us. And our contact information is on the Genomic Vision website. So it's genomicvision.com.
Awesome. Brilliant. Thank you very much, guys. Absolute pleasure. And good luck, and I hope you will see some successes.